Okay, hi everyone, my name is Ángel, so let's see a bit, and I'm going to talk about, well, I'm a PhD student I'm a PhD student, sorry, in iuFOR, and I'm going to talk about water stress response in Scots Pine. So, as all of you know, global climate is changing. So there is an increased likelihood of extreme events, a change in precipitation patterns, and a rise of mean global temperatures. These changes are causing too many species fail to adapt to the new climate conditions and are causing many die-off events worldwide. Here we have two Pinus silvestris or Scots pine populations, one in Switzerland and the other one here in Almeria, that are in decline situation. And Scots pine is a really dominant forest species in the Arctic region, but it's very susceptible to these changes due to its water and temperature requirements. It's already known how plants suffer with water stress. First of all, plants stops their growth, the net carbon balance fixation decrease photosynthesis values, and ultimately cells lose turgor and die. So our main objective is to define point of drought stress which Scots pine doesn't recover its normal metabolic and physiological functionality, and to reach that objective we want to combine ecophysiological measurements and a metabolomic approach. So to reach that objective, we did a common garden experiment with 3-year-old Scots pine individuals that were grown in a greenhouse for at 55% of humidity between 10 and 25 degrees. So this is our experiment outlined. So we have treatments of drought stress from 10 to 60 days in steps of 10 days. Controls were always well watered, and we took ecophysiological measurements and needles and root samples for metabolomic studies in the end of each treatment. To assess the capacity of recovery of these plants, we repeat the same sampling 1 and 2 months after each treatment. So, as a really general overview of our preliminary results, well, I will talk only of physiological results. We did a PCA, which is a dimensionality ordination of is a multivariate ordination that relates our variables and our samples. Each shape is one sample, and controls are circles, and treatment samples are triangles. The included variables in this PCA are the water potentials measured at pre-dawn and at midday, the net carbon assimilation rate for the plant, the stomatal conductance for the leaf, the internal concentration of carbon into each leaf, and the cell damage produced by drought stress and measured by the ion leakage of intracellular ions. So here we can see two clusters that separate along the epigeneal component 1 that explains the 65% of variability, being controls in having higher values of water potential, net carbon assimilation, and stomatal conductance, and the drought treatment individuals higher values of cell damage. So here we can see that the plants that suffer only 10 days of drought stress are quite similar to controls, and the other treatments, it's going along this PC1. So here in the upper right corner, we have two individuals that are basically outliers, and we think that they were dead in the time of measure, so they will be excluded for the next analysis. So here we have a box plot that represents the water potentials measured at pre-dawn and its treatment of the experiment, having higher values of water potentials in control individuals, as is expected, and the water potential decreasing along the experiment is going on, and the water stress is more severe. Here we can see how the plant more or less can maintain their water status until day 30 or 40 of treatment, but later water potentials comes down. In the right, we can see different treatment individuals, and we can see how 10 days drug treatment individual has no visible symptoms of drought damage, but when we are going into a more severe drug treatment at 30 or 60 days, we can see the symptoms. When we correlate the net assimilation of carbon dioxide with this water potential measured at pre-dawn, we can see a sharp decrease and a very low, at very high potential, at being 0 at more or less 1.5, 1.7, minus 1.5, 1.7 megapascals. Later this point, the net carbon fixing is 0, so the plant is breathing more than they are fixing. They are producing a carbon dioxide, like all of us. In correlation with that, if we correlate these stomatal conductance with the water potential measured at pre-dawn, we can see how the plants close their stomachs at more or less 0.8 megapascal. So they are closing very early their stomachs to preserve water at doing a bit of photosynthesis. Another way to assess this damage produced by a water stress later of produced in the cell, we can plot the ion leakage of intracellular ions with the net photosynthesis. And we can see how very low values of cellular damage we can see this decrease. And when cellular damage is visible, you can see the net carbon intake is negative. So our preliminary conclusion is that a Scots pine individual can maintain their water status until day 40 without watering, and at values of more or less minus 2 megapascals, the net assimilation of carbon dioxide is 0. So the plant is or breathing or dead. So our next steps is analyze the metabolic strategies or pathways used by Scots pine to cope with this water stress, focusing on certain metabolites that we know that are playing these roles to protect the plant, and they assess this resistance capacity of Scots pine 1 and 2 months after the treatment, using both the ecophysiological measurements and the metabolomic pathways. So thanks for your attention, and the autonomic organism of national parks to the foundation. Thank you so much.